This vaccine is among the 8 that are ahead in terms of being tested in humans for efficacy. A high-profile potential vaccine for COVID-19 being tested by researchers at Oxord University did not protect vaccinated monkeys from being infected by the virus. However, the test animals seem to be shielded from pneumonia. Also read, WHO backs COVID-19 vaccine trials that deliberately infect participants. The vaccine candidate, AdOx1-nCoV-19, being tested may be a weakened form of standard cold virus, adenovirus, that affects chimpanzees but has been neutered to stop replication in humans. Advertising reports of the candidate vaccine's performance in monkeys, rhesus macaque, have prompted researchers to check the vaccine's potency in humans. Its promise has also led to Indian vaccine manufacturer, the Pune-based Serum Institute, announcing plans to manufacture a 4 to 5 million doses by end May in India. It is one among 7 global institutions which will manufacture the vaccine being developed by the Oxford Vaccine Group. However, detailed results of the trials in monkeys available on pre-print server bioRxiv suggest that, supported these results, the vaccine might not be the panacea to protecting people from being infected and spending on the infection to others. The research paper is yet to be peer-reviewed. Also read, coronavirus, around 30 Indian attempts at COVID-19 vaccine, says Principal Scientific Advisor Rajesh Gokul, Faculty, National Institute of Immunology and former head of the CSIR Institute of Genomics and Integrative Biology, who has read the paper, said that in an ideal world, no company would continue testing the vaccine in humans. Supported the available data in monkeys. We have presence of the virus within the upper tract of the animals. It is possible that these can come down again to the lower tract and cause pneumonia. Ideally, if you've been inoculated with the vaccine, you ought to be ready to substantially filter out the virus, he told The Hindu. The researchers, within the IR paper, acknowledge the presence of virus in the upper tract. Despite this marked difference in virus replication within the lungs, reduction in viral shedding from the nose wasn't observed, they note. Also read, coronavirus, when will a COVID-19 vaccine be ready? They explain it as being possibly thanks to the unusually high amount of the virus that the monkeys were exposed to. Unusual, therein citizenry were unlikely to be ordinarily exposed to those quantities of the virus. The researchers, led by Sarah Gilbert of the Jenner Institute, University of Oxford and Vincent Munster of the National Institutes of Health, argue that the presence of virus was significantly reduced in bowel fluid collected from the lungs and lung tissue of vaccinated animals than within the animals that weren't vaccinated. Moreover, virus-specific neutralizing antibodies were detected in those macaques vaccinated and no such antibodies were seen in people who didn't get the vaccine. For their analysis, they vaccinated 6 monkeys with the candidate vaccine and 3 got a control vaccine called AdOx1-GFP. Based on these results, 1,110 people are participating in human trial, half receiving the vaccine and therefore the spouse, the control group, receiving a meningitis vaccine. The dose of the vaccine was half that of what's getting used for humans immediately. This vaccine is among the 8 that are ahead in terms of being tested in humans for efficacy.